Hi everyone, welcome to Foresight's Biotech and Health Extension Group, sponsored by 100 Plus Capital. We are really picking up steam. We have a variety of really fantastic keynotes coming up. I'm super happy to today be joined by Morgan Levine and Jamie Justice to discuss biomarker standardization. I won't say very much about it. I think they can do that much better than I can. I'm really happy to welcome Morgan here again for the second time now after her first presentation. I've had a plethora of emails of wanting you back on, so I'm really happy that you were able to make it. Thank you very much. And Jamie, I'm super happy that you're here for the first time presenting, and I think the presentations are I think going to be quite nicely building up on top of each other. So thank you both so much for joining. I really appreciate it. Two things up front. Number one is that if you haven't done so already, please sign up to our swap card. This allows you to schedule meetings after this meeting, so personal one-on-ones. So the main benefit of the platform is that it allows to take me out of the collaboration group loop, so that I'm not bottlenecking collaboration anymore. That may be useful to some of you. So please sign up to the platform, and Morgan and Jamie have both agreed to stay 15 minutes longer so that you can book one-on-ones with them. So really cool, thank you. And then secondly, we also have a code for Elysium. So Morgan was able to put me in touch with Elysium, and so for those of you who are interested in the Elysium index test, we now have a code for people that are members of this group to get a discount there. So if you're interested in that, then please also go on our member benefits section on the swap card area. And if you like Morgan's talk, and if you're excited about the things that she's discussing, then perhaps this test is of interest to you. Anyway, I'm super happy to have Morgan and Jamie both here. Thank you so much for joining. I'm looking forward to the discussion and to the one-on-ones afterwards. All right, without further ado, Morgan, please take it away.
Thanks, Allison. I'm really excited to be back talking to this group. And actually, the two things I'm going to talk about today are brand new. One of them I've never even shown. So, and it's not published, so I'm giving you guys kind of a sneak peek. The other one's under review. Okay, so today I'm going to talk about quantifying aging, mainly using DNA methylation biomarkers, because this is mostly what we work on. And to jump right in, because I know we don't have a lot of time, to me, actually being able to quantify the aging process, I think, is one of the most important things right now in the field. There's a lot of emphasis on, can we actually intervene in aging and either reverse or slow it, but I would argue that there's no way to actually definitively test if you did that, if you can't directly measure the thing that you're trying to intervene in. So a lot of people think, at least in the general public, when you say, what is aging, they think of chronological aging. And really that's because chronological time is correlated with actually this biological aging process. So these, however you define this, but changes that really alter a living system, and how we define it is just makes it more vulnerable to failure and decline. But we know that unlike chronological time, the rate of biological aging is malleable. So it doesn't tick at a constant rate. We can see this based on between species differences, within species differences. So we actually think this, if we can quantify this and distinguish it from chronological age, this will actually give us exciting tools that can help us kind of in these three areas. So first, they'll provide clinical trial endpoints, which I won't go into too much because I'm sure Jamie is going to really go into that. But essentially, it'll give us endpoints to actually measure whether we've slowed or reversed the aging process. Often, in mouse studies or other model organisms, they'll use lifespan, but in human populations, this really isn't feasible. It also will inform us on basic biology, so understanding the mechanisms that either drive the ticking of biological aging or modulate it. And then finally, it can be used for understanding risk stratification. This can be on a personal level for people who like to do kind of quantified self and actually understand their own risks and how to change those, but also on a population level to inform things like policy or secondary prevention. All right, let's see. Okay. So as I mentioned, I'm going to talk about epigenetic aging. People have probably heard about epigenetic clocks, but just to give kind of a brief rundown of what we actually mean when we talk about this. is that we actually think, so the epigenetic pattern in a cell is really what's dictating the cellular state, the cellular functioning. So most of your cells, aside from somatic mutations, have the same DNA, but what makes a stem cell, or skin cell is really the epigenetic pattern. And this is really kind of personified in Conrad Waddington's Landscape, where he talks about how with epigenetic changes, you can get an undifferentiated cell that will basically traverse this landscape and differentiate into different cell types that have different epigenetic patterns. But aside from kind of differentiation in cell state and cell identity, we actually think, and people have shown, that the epigenetic pattern can also distinguish young cells from old cells. So this is really where we're interested. And for this, we measure DNA methylation, which is just one form of an epigenetic modification. So briefly, it takes place at these what are called CpG dinucleotides. And basically, the cytosines can become methylated. And what we see in aging is not just a change of, not just increases in methylation or decreases, but really a change in the pattern. So you'll have some of these CpG sites that tend to show declines in methylation with aging. And this is really measuring the proportion of cells in the sample that have methylation at this site. So for instance, you might have a sample from a 20-year-old where you have 90% methylation here. And then by the time you look at 80-year-olds, you'll have on average 60%. You'll have others that actually increase or become more what we call hypermethylated with aging. So perhaps 5% in a young individual, and this goes to 45%. These two are kind of what we consider epigenetic drift. So it's moving towards 50%, where you kind of have a random 50-50 split in the cell population. But we also see sites that change kind of away from the mean. So not a regression towards the mean, where you might start with relatively low methylation or relatively high and actually move more towards the extremes with age. So because this has been so striking, people have developed these epigenetic clocks. And really what they are is they're basically saying we can look at a sample and using some algorithms, say based on the pattern at specific sites that we are measuring, can we predict out the age of the sample? So the first clock was developed in 2011. I won't go through all these clocks for the sake of time. The clocks up until our clock in 2018 were all developed as predictors of chronological age. So we usually refer to these clocks as what we call first generation clocks. So they were trained as chronological age predictors. However, then we and then others also showed that you can make these clocks that are actually much more robust in terms of predicting things like mortality or disease incidence, trained on aging correlates. So that's kind of what has been done more recently and where the field is going. So in terms of thinking like what the goals should be of having a good aging biomarker, let's see if there we go. So number one, it should have good prediction. And this is prediction of chronological age, because of course you need a biomarker that tracks with age to say it's actually an aging biomarker. It shouldn't be a perfect prediction of chronological age because, again, chronological age is imperfect proxy of this biological aging process we're trying to capture. And for prediction, we also wanted to be able to predict outcomes above and beyond chronological age. So looking at people the same chronological age, will it differentiate people who are at greater risk of mortality, who are at greater risk of disease incidence, et cetera. The other thing is it should have high precision, in my opinion. So I'll talk a lot about this in a minute, but basically, if I measure the same sample twice, am I going to get the same answer? That's really important for people who are measuring these, who are tracking them in themselves, or it's really important for things like clinical trials, maybe less so for kind of large population data or epidemiological data. But in terms of what we're mostly working on, the precision becomes really important. Obviously, it should be non-invasive. This is a problem when we're talking about measuring aging in tissues that are actually not accessible. Preferably, you want something that's modifiable, because if it's not modifiable, I would argue, why do you need to know the epigenetic age of something? And then finally, working on things to make it more affordable. So I'll touch upon a little bit of each of these, maybe not so much the affordability and modifiability, but that's really the goal. All right, so talking about precision and reliability. So basically what I mean by this is if I split a single blood sample, so I take one blood sample from someone at a single time point and split it, and I assess the epigenetic age on that sample twice, will I get the same answer? Or will I get one of the samples says that person has an epigenetic age of 50, and the other one says it's 55. And you can imagine why this might be a problem for clinical trials or personal tracking. So actually about 2 years ago, when I actually started working with Elysium, this was a question that came up because obviously if you're going to offer a direct-to-consumer test, you want to make sure you're giving people an answer that you can stand behind. And I discovered pretty quickly that actually the existing epigenetic clocks are fairly bad when it comes to this. So these are samples, they're not in different batches, they're literally measured on the exact same array, the same blood sample, and you can get differences. 5 years, in this case, for pheno-age, it's actually really bad. You get up to 9 years difference in the exact same sample. Grim-age is actually one of the better ones. This is partly because Grim-age includes chronological age and gender, which obviously in replicates is going to be a constant. But you still get some deviation of about 2.5 years in Grim-age. So before putting out index that Elysium offered, I did a lot of work on actually trying to figure out how to deal with this. And then since then, my postdoc, Albert Higgins-Chen, has been working on this. So I'm going to talk about what he's kind of done for a paper that's currently under review. So the reason this happens is because actually a lot of the CPGs that we measure on these arrays are very noisy. So this is the ICC, which basically you can say kind of how much agreement you get between replicates. And you have actually a lot of them that are very poor. And this kind of shows the average methylation level and as a function colored by this ICC. So you can see CPGs that are very hypermethylated, so tend to be towards fully methylated across all the cells in your sample, or hypomethylated, also tend to be noisy. So there's actually some biological explanation for this. It's also because these CPGs don't vary that much. So a certain amount of noise is going to seem like a big deal in CPGs that have low variance. So, you know, one thing that people might say is, okay, well, what if we just exclude noisy CPGs and then remake clocks? So we tried to do this first. This shows how much we did pheno-age here just because it was the noisiest, showing how much it deviates if you do this. And really, you can get all the way up to 0.9, and you actually don't improve this kind of replicate reliability that much. But what you do lose is your ability to predict things. So if you're cutting off at 0.9, you get this sudden decrease in your ability to predict mortality. So this is obviously not the right way to go. And actually, this suggests that even these noisy CPGs are actually really biologically meaningful. So the solution we came up with is, can you actually capture more higher order patterns across the genome? So if you think about this, CPGs are highly correlated, and really what the clocks are doing are picking out one CPG to represent some signal that actually a lot of them are sharing. So we pick this CPG, but really the signal's captured by all of them. So what we wanted to do is actually say, well, what if we looked at all of them to get a more reliable signal, right? Maybe one of the CPGs has some error in it, but if you look at the group, you're going to actually do a much better job. The group shouldn't be picking up this kind of random noise. So that's what we did. We have the paper on bioRxiv, it'll hopefully be out soon. So if you want to go into the statistical details, which I won't do today, feel free to check out the paper. But this is what it looks like for after we apply this. So these gray dots are the original pheno-age, and you can see they're off this kind of zero intercept slope of 1, but you get really good, almost perfect correlation after you apply this method. And it doesn't just apply to the pheno-age clock. And again, here you get deviations of 9. We bring them down to most of them, all of them are under 2, most under 1. And this happens across the board for all the clocks we tested. So the gray is the ICCs for the clocks in their original form. The blue is for them after we apply this method. So you're getting the same clocks, just much more precise. And then the important thing is, okay, well, did we do anything to actually hurt our ability to predict outcomes? Here I'm just showing mortality, but we looked at tons of different outcomes. What you can see, the blue again is the new method, the gray is the original. Even Grim-age, which is highly predictive of mortality, improves a little bit using this method. So we don't actually lose our ability to predict. If anything, we actually gain some predictive ability. This shows longitudinal tracking of either the original clocks here and here, or these corrected clocks. And as you can see, there's a lot less jumping around. Some of these are really clean in terms of longitudinal tracking, so they should help with that kind of data. And then the other thing that, again, we're really interested in is how would this impact using clocks for testing interventions? So this is kind of the idea that you'll have a placebo and an intervention for people who are, let's say, chronologically 50 years old. You follow them up a year, and maybe they only deviate now by 1 year epigenetically. Can you pick that up using the original clocks that are noisy, or versus picking them up using these new clocks? And Albert did these power calculations that show for a given effect size, so how much you think your intervention's going to change epigenetic aging, what the sample size you would need to differentiate your case control using in the gray, the original clock, and blue, the new clocks. So this is for all these different clocks. You can see that the blue one is much lower. So you actually hugely increase your power. So you need much fewer samples than you would have otherwise needed had you used the original clocks. And we did this, imagining it in an older population or even a younger population that has less deviation in epigenetic age already. And then we're also really interested in vitro studies of epigenetic aging, and we're doing a lot of those in-house. So we just took some of our data we had from there. This is human astrocytes that we serially passaged. You can see that using the original clocks, which is this top row, you get lots of jumping around, not this very clear pattern. Using these new clocks, you get really precise, and actually you can hardly tell the difference between the technical or the biological replicates until about passage 8, when they actually start diverging. We actually think this is biologically real, and it's not actually just something to do with noise. Again, we looked at kind of this power analysis for if you want to do drug screenings in vitro experiments using either the original versus the new clocks. And again, we find that you need much fewer samples to do this with the new clocks. All right, so that is all I have on that section. And then this is where I'm going to briefly just talk about a brand new clock that we're almost actually, it's not even completed, but I wanted to share it with you guys. It doesn't have a name yet, so if people have a good name, feel free to let me know. Right now, we're just calling it a system clock. So in an ideal world, you would actually be able to non-invasively get measurements of aging in different physiological systems or organs, right? We would want a brain age, a kidney age, a liver age. In reality, most of these samples require biopsy. Often they're done post-mortem, so you're not going to be able to track these easily. So this is kind of what we tried to do. The nice thing is our PhenoAge, which some of you are familiar, demonstrated that we can actually capture kind of multi-system composite aging measures using DNA methylation in blood. So we actually think that the signal's in there, you just kind of have to pull it out. So our goal here was to build system-specific measures that when you combine them, you can actually get a robust overall aging signal. So what we did is we combined first clinical biomarkers, trait variables, using unsupervised machine learning approaches. So we kind of designated them to a different system or organ group, use unsupervised machine learning to find kind of patterns in that group. And then we use supervised learning to train a methylation predictor of those patterns. So we got scores for what we call brain or cognitive, inflammation and cytokines, immune, which is leukocyte measures, a metabolic one, hormonal, kidney, liver, cardiovascular, and like a red blood cell / platelet measure. And I'll just briefly show kind of how the full system one compares. I'm not going to go too much into the individual ones, but you can pull them out and look at them individually. So we compared them against just 3 of the existing epigenetic clocks. So this is for all-cause mortality, and you can see it's substantially better predictor of all-cause mortality than existing clocks, much better predictor of cognitive functioning than any of the existing clocks. And we think that's really being driven by the fact that we have this brain cognitive component to it. Much better predictor of physical functioning and also comorbidity counts. And we have other outcomes too. And to borrow this term from Mike Snyder, this ageotypes term, the nice thing about this is you can pull apart the systems and actually look at profiles of people. So you might have two people that have the exact same full system age, but one of them might get that because they have accelerated liver aging. Another one might get that way because they have accelerated in some other system. So we can actually define different subtypes or ageotypes or whatever term you want to use for people. We actually think this will help in potentially in intervention trials because you might not want the same intervention for everyone depending on kind of what their subtype is. So finally, kind of as what Allison mentioned, I just quickly want to talk about kind of this critical next steps I think need to happen in the field. For me, the most disappointing thing right now in the epigenetic clock literature is our lack of mechanistic understanding. This is really where my lab is interested in moving more towards. So really understanding, number one, what is it that drives epigenetic aging, or what is it that we're actually picking up? And then how is that actually linked to the phenotypes or manifestations of aging that we can actually predict using clocks? So we're trying 3 different approaches to get at this. So we're doing, as I mentioned, a lot of in vitro analyses. We're also doing a lot to try and look at single cell DNA methylation and really understand how this pattern plays into kind of cellular heterogeneity and shifts in cell populations with aging. And then finally, we're using computational approaches to kind of what we call kind of break apart the clocks and figure out what the different components are that give way to the overall clock score. We think that the different components will actually have different kind of biological mechanisms. You can't study the clocks as a whole for a mechanism. And finally, I just want to acknowledge all the people in my lab at Yale. I couldn't list all my collaborators, but just some of my collaborators elsewhere in my funding from NIA and Glenn Foundation. And then Allison, are we doing questions now or after Jamie?
I would say we go with Jamie because then we can have this like an all-on discussion. Thank you so much, Morgan. I just shared your health extension challenge in the chat here, so people can look it up and also see what other keynote speakers have produced as challenges. Thank you, Morgan. This is fantastic. I think you already have a suggestion for a name, which is OmniAge here in the chat. So maybe you can answer that and see how you like it. Okay, thank you so much, Morgan. I really appreciate it. And maybe you can already answer a few questions in the chat, and then we'll move to a discussion after Jamie's presentation. Jamie, thank you so much for coming. We're really excited to have you. Yeah, I'm super excited about your presentation. I think Morgan has already, paved the way and gave a really good introduction for why people may care. And so thanks a lot. The stage is yours, and we'll move to discussion afterwards.
Okay, great. Thank you. So I'm Jamie Justice, and that is, it's quite an act to follow, Morgan, always intimidating to share after you. I want to thank you all so much for hosting me virtually, and a special thanks to you, Allison, for your tireless efforts to organize and herd all of us cats. It looks like my screen may be freezing a little bit. We have a hurricane going through nearby where I live in North Carolina. Hopefully I maintain internet connection. So I apologize if I phase out a little bit. All right, so I am going to give a talk that's a little bit different. And for here, it's actually, I'm going to be focusing more on my work is around trials. And so I like clinical trials, I work in them, and really what I'm interested in is applying and use case for biomarkers in clinical trials. Okay, so I have nothing to disclose. Hopefully most who are here have some familiarity with geroscience and the pillars or hallmarks of aging, and might be familiar that there are a number of promising interventions that can target those processes. So what I'm showing here, these are just a flavor of various behavioral, dietary, and pharmacologic interventions that are now being advanced to clinical trials. And so as we are thinking about moving these to trials, we have to develop trials in order to test them and create frameworks for how we should do that. So this is a translational research pipeline for drug development, and it's a really arduous process. So the timeline is well over a decade from discovery to FDA approval, and very few drugs make it through the gauntlet of rigorous testing. So the estimates that are shown up here, these are actually, these are for relatively traditional linear disease pathways that have generally accepted outcomes to weigh success versus failure. As most on here know, that's not the case for aging. I'm getting a Max Headroom thing going on with my screen. Hopefully my internet connection holds out. I apologize for those listening at home. Maybe there's a way we can correct this going forward. All right, and so, so this would be the normal pathway that doesn't exist for aging. So in the absence of really clear regulatory pathways, I've been fortunate to work within 2 different paradigms for moving treatments into clinical trials. And this includes various disease-focused trials and larger prevention trials. And so in one, we might take a single investigation or investigational agent or treatment, and we begin testing it in different disease cases on an individual basis. And so then investigators would actually be testing this sort of in different disease silos and then applying to FDA to get an indication for each one of these diseases individually. And, it's not great, but it actually is still is a good application of geroscience, is that it provides new treatment options for hard to treat diseases. But one of the greater hopes is that if we really harmonize some of the approaches and we can look collectively across these diseases, then eventually what we begin to observe may converge and make a mosaic that looks a lot like aging. And this is something that having a harmonized set of biomarkers and a standardized process to implement these becomes really critically important in taking these to sort of the next stage. There's also an alternative approach that we've been working hard on, and this is to consider really what an aging outcome should be and to design a trial with sufficient power to test that. And so in thinking about like what that aging outcome is that it has to balance our collective scientific understanding of what aging is or look like, those ageotypes. I love that so much. I was really looking forward to Mike Snyder's talk today and was disappointed that he wasn't here to join us. But, it's also very important that when we're thinking about this, it has to communicate back to the FDA in the US, so that we need to try to find an intervention that changes the way an organism feels, functions, and survives. And so I would argue that both of these approaches are really critical to push progress forward. And I've been afforded the opportunity to work on both. And so there are key clinical trial design elements that are common to both. Certainly, we're testing interventions, we have different populations, and of course the outcomes and biomarkers share a certain flavor. Within the interventions, the critical component is that we're looking at interventions that target critical aging pathways. There are some differences, certainly with treatment trials, is that they have a certain tolerance for higher risk, newer or repurposed agents, whereas prevention trials tend to have a more established safety record. Populations obviously would differ depending on existence or absence of disease. Really for the outcomes and the biomarkers, I'm going to put these both up, together, is that, I think they can be considered along the same continuum. And I'm going to come back to this continuum idea in a little bit. But when we're thinking about these, I think of them like Russian nesting dolls for some of the outcomes, especially for the treatment trials, is that you might be doing this under a disease condition. But ultimately, what we're really interested in, whether we're talking about biomarkers, or we're talking about outcomes, is that a lot of us, even though you're looking at this in a disease condition, it's like, we're really interested in sort of the next layer. And so what are sort of the geriatric facing or the aging facing outcomes and biomarkers that can really harmonize? Okay, my connection is getting really unstable.
Maybe
we can hear you.
Yeah, we can hear you well. I can hear you.
Okay, great. Okay, very good.
If it gets more unstable, maybe you can cut the video.
Okay, we might have to, because I keep getting, I'm getting warning signs on my computer. So I'm glad you're not seeing those.
No, it's great for us.
Okay, good. And so I think the biggest underlying thing here when we talk about biomarkers for trials is that this is if you take nothing back from my talk, it's this, is that there is no perfect biomarker, but some biomarkers are useful. And really what it comes down to is the context of use. All right. And so now, again, another step backwards. I know that we just had recently one of our FDA officials talking about biomarker qualification. It was a really fantastic talk for anybody who might have missed it. So starting with sort of a centering definition, biomarker, it's an objective measurement that reflects the interaction between the biologic system and a potential hazard. And so in the FDA language, some of the things they talk about for qualification kind of divide these two, right? That you're looking at something that might be an indicator of a normal or pathogenic process, or it can be sort of this measure as a response to intervention. But the way that a lot of us thinking about it with trials are really considering this is the emphasis is on the interaction of the two, is that it's not good enough to have one or the other. It's really how they play together, testing the biology, but looking also at the response to an intervention. And again, going back to some of these FDA language, just again, it's a nice centering thing. For those of you who missed it, I'm sure Allison can show you where to find it online. So these are some glossary terms for different biomarker classes or context of use that are really critical. For trials, ones that I'm considering most often are things about monitoring. So looking for things that can assess status through serial measurement. That's really important for trials. Also safety, of course, is looking for the likelihood, presence, or severity of toxicity of different agents that we might be testing. And then what I consider the holy grail really is, looking at the pharmacodynamic or response to intervention. And so these are things that can actually look at the biologic response in a person who's received treatment. It could be sort of on target, whatever is really specifically being targeted with that agent, or it might be more collectively looking sort of at larger aging phenotypes, what kind of markers might be present there. It's not that these other uses are not important. Again, they're critically important, but it would really depend on the context of use. For example, if you're doing a treatment trial, looking at diagnostic or susceptibility of risk might not be quite as important because the disease would already be present. However, there are some contexts in which this does become important for trials. And so I'm going to finally get into a couple of example cases after all that information. The first are trials on xenotherapeutics. So this is a really great example of that mosaic approach that I talked about before, about testing one agent in a number of different conditions. And so this figure shows that really well. In addition to being what I think is maybe one of the most terrifying digital images of a human, it actually gives really great content to show the number of different conditions that xenotherapeutics and Senolytics are being tested in. So all of these, this is just published in 2020. It's already grossly outdated. I think at my last count, there's between 20 or maybe upwards of 27 different interventions being tested that are listed on clinicaltrials.gov. I have one of these starred here for dasatinib and quercetin, which in idiopathic pulmonary fibrosis, which is a use case of the Translational Geroscience Network that I was fortunate enough to lead. And I want to highlight a couple of things with this trial. One, IPF, idiopathic pulmonary fibrosis. It's a quintessential disease of aging. It's ultimately fatal, and it has an estimated survival of only 3 to 4 years after diagnosis, and there are very few available treatments. And though the etiology of this disease is really poorly understood, converging evidence suggests that the aging process, cellular senescence, may be at the nexus, a central contributing mechanism in this disease. And so some of the foundational data in this regard was shown here. This is by a couple of rock stars in our field, Nathan Labrasher and Marissa Schaefer, which if you have not invited them to give a talk yet, shame on you, put them on your list. And so, they really did do some of the really great work looking at markers of senescence in this disease. And this became hugely foundational. They found that not only were some of these senescent cells identified by P16 positive within the lung and fibroblast and honeycomb structures, but they also found that expression of certain senescence markers and and expression of senescence-associated secretory products also were elevated with increasing IPF disease severity. And so that was important, plus some of their preclinical work in disease models showing possible use of things like dasatinib and quercetin in that disease context. And so that was the foundational evidence that we used to launch the first trial of Senolytics. And we did this in persons who had stable idiopathic pulmonary fibrosis with an age criteria. I'm highlighting this because we did not have a biomarker available that was standardized or validated to say whether or not the people that are enrolling in this trial had senescent cell burden that was elevated at the time of trial entry. We didn't have it because it doesn't exist. So this is one of those context of use things that, you know, we're going on diseases that known to have some kind of senescence associated feature based on foundational evidence. And then we move into trials without actually screening or gating for entry yet, because simply it doesn't exist. And the reason it doesn't exist is, you know, one, it's development, and two is that, you know, there are a lot of efforts ongoing in order to try to move that forward. And so anyway, we enrolled these folks, and what was published is our first open label trial of dasatinib and quercetin. It was a short 3-week intermittent dosing trial. This was followed up by work that was run exclusively with our colleagues at Texas, that they did a randomized control trial. And this was really a feasibility tolerability kind of study. And so, you know, we're looking at what different assessments we could possibly do in these people, dosing strategies, and really important, you know, what kind of functional measures can they tolerate? What outcomes, patient-reported outcomes can they contribute? But really central to this effort was, are there biomarkers that we can use in the context of a trial? This was sort of the Wild West of doing work in senescence, and there simply weren't any that were accessible. And so, you know, long story short, you know, we found some interesting things in the trial. The ones that have been, that, you know, we're using to drive sort of next stage, we're actually planning for an efficacy trial now. And those are really because we saw that there were some improvements in mobility. And we saw that there were clinically meaningful improvements in mobility in the absence of pulmonary specific measures. And really importantly, in the absence of really solid biomarkers. And so the take home point is that there were no withdrawals, even though there were some anticipated side effects that were seen. There's some potential improvements in functional measures that are common to geriatric assessment, but not disease specific measures. And the biggest point for me was that we didn't have, at the time of doing that trial, anything that really could comment on the pharmacodynamic response. So, right, we didn't know exactly what the senolytic effect was. Did we reduce burden? And we had a few insights from circulating markers, but, you know, back to this point is, you know, standardized, validated biomarkers are absolutely essential to driving next stage trials. And the other thing that we were still working on is how do we integrate those sort of target specific biomarkers with more general biomarkers of aging to really dig into sort of that ageotype that Morgan did a great job describing. And so again, the efficacy trials are warranted, but we really need aging outcomes and biomarkers. And there's also another caveat here, is that when doing trial design, we also have to sort of accept the risk of potentially a failed trial on some disease specific endpoint, as even if it means that you might have a meaningful change in some kind of aging outcome and vice versa. Okay, so back to biomarkers. There have now been some efforts in this world to sort of show follow-up to that there might be some movement in biomarkers of senescence with Senolytics. This was a follow-up trial looking at P16 and P21, 2 biomarkers of senescence in whole adipose tissue before and after a single 3-day course of Senolytics. And there does seem to be some movement there. But again, this requires, you know, invasive use of tissue, and the other half of my world is actually looking at senescence markers in adipose tissue. And I can just tell you there's still a lot of bugs to work out. And that's why it's really important, is that there's a related concept. This is not a biomarker thing, but there's another effort within the NIH Common Fund is it will be funding these mapping centers. So this really gets down into that single cell level, and that we can really begin to characterize what senescent cells look like in healthy human tissue. And I'm really careful to say that this is not a biomarker project, but this is the foundational evidence that's necessary for us to be able to build, validate, and standardize biomarkers of senescence for trials. And so that's important at the tissue level. And there's a lot of other folks doing work, again, looking at this and linking it back to biomarkers that might be able to access in the in circulating cells. Once again, my two favorite humans, Marissa Schaefer and Nathan, they did this work looking at, they induced senescence, measured secreted factors, and began to test these under different clinical conditions. Coming down to a panel of SAS factors that they found that had been secreted and could be measured fairly reliably in circulating, I think it was plasma or serum, one of the other. And what I wanted to highlight with this is that what I found really interesting, and is another great point for discussion for this group, is that, we're talking about many of these factors as if they're sort of senescence associated. But I think a number of people, I heard some people, and when we were discussing before, might be doing work in proteomics. There are others that are interested in other kind of biomarkers, is that a number of these factors aren't necessarily senescence associated only. That there is sort of this larger sort of aging connection that we're sort of looking at more of a general milieu that also encompasses senescence. And so I think that carries us into sort of, if there's time for it, spend a couple of minutes talking about how this relates to the alternative strategy, the one that's absolutely imperative that we make progress on if we're going to move any of these areas forward. And that's actually developing an aging outcome that can be recognized by regulatory officials. And so this has been work that I know Nir was on here, and I am so grateful to Nir and many others that as a postdoc, I was brought in about 6 years ago to be the underling, the minion for this development of this trial called TAME, Targeting Aging with Metformin. They've kept me on, and I'm now have the opportunity to serve on the executive committee for this trial, and I'm in charge of trying to develop or coordinate whatever the biomarker and biorepository strategy is for this trial. So for those who don't know, TAME is Targeting Aging with Metformin. It's a 6-year double-blind randomized placebo-controlled trial that will be conducted in, I think now over 3,000 non-diabetic adults and 14 clinical sites. And most importantly, it's the first trial that was designed, really crowdsourced designed by a group of scientists across the US to create a regulatory path for clinical trials to target age-related multimorbidity. And when we think about what that aging outcome would be, this again becomes really critically important, is that all of these outcomes, they exist on a continuum and they're not exclusive. So we have, as we go across this continuum, we have biomarkers and other proof of concept things on one end, and on the other end, we have things like, median lifespan, life extension, other delayed frailty and age-related diseases, and the increase in time, expense, and salience required to see that outcome as we go along. Really important is FDA, if we're looking at something that can be properly adjudicated for a trial, then we're talking about things that are out here on this end. So it's not that biomarkers are not important, it's just simply that for an aging outcomes trial that reads for the FDA is that we have to change function, feel, survives first. And then we can actually have something that we can go back and begin to validate and standardize biomarkers against. Okay, so looking at that, if we have an FDA-facing outcome, then if it's an effect as on aging, then we should be able to reduce the incidence of multiple diseases or geriatric syndromes, and those diseases should share few risk factors other than chronologic age. And so looking at that, the outcome then becomes this time to incidence of one of a collection of possible diseases or endpoints and age-related conditions. And then we have, again, back to this Russian nesting doll thing. So that's right, that talks to the FDA. The part that talks to a lot of us that are actually doing this work is this sort of second effect, is that if the drug's effect on aging, is that in addition to that, is that it should improve or attenuate declines in functional outcomes and clinical phenotypes of aging that are important for persons who are aging, and also communicates well to the clinical community. We also need to have a drug that can affect and improve biomarkers of aging in aggregate that really reflect that underlying biology and creates a platform that gives back to the scientific community to drive next stage discovery. And so that was really the concept that went into designing the TAME trial, which again is a 6-year trial looking at a clinical outcome that talks to the FDA. And then we have these other outcomes that we think are actually, I don't know, I think they touch me more as we talk about function and biomarkers. And, you know, and thinking about this, going back again to this best category thing, as we go back to these 3 that are just really critically important. We know we have to monitor, we have to look at safety, and again, the holy grail of pharmacodynamic and response. And so we need the drug's effect on some of the targeted processes that we expect it to target, and really to reflect the change in the underlying biology. And very important to all of this, especially when we're looking at some of these really large scale trials and some of these first kind of trials, is that it's incredibly important that we create a harmonized resource to the scientific community that more people can use to build. And so in doing this, I don't have time to go into any of the details. I chose to keep it kind of broad today, that we can look at this sort of collectively across different platforms. We think about these, you know, that we could have some biomarkers of aging that really reflect some of those pillars or hallmarks. A lot of those aren't validated or standardized yet. Morgan gave you a great explanation of what's happening with methylation-based biomarkers, and we have been really fortunate in the TAME trial to, you know, we've hooked Morgan in and have asked her opinions and work, and there are so many people that are working in this space that I really do believe that as far as what we have developed right now, in the time since we began developing this a couple of years ago, I was a naysayer, but where the field has really come in the last couple of years is that some of those methylation biomarkers, they really are becoming some of our most advanced biomarkers for trials. We can't validate them yet because it requires a trial for validation and qualification. But these are at least things that we would have the platform to be able to do some, you know, larger 850,000 CPG sites so that we could begin to validate many of these methylation biomarkers. And of course, there's going to be a lot of work into looking at deficit and damage accumulation, whether these are from, you know, deficit accumulation just from clinical functional safety measures, or if we're beginning to look more broadly at maybe proteomics or other sort of larger based platforms so that we can begin to marry some of these ideas back together. And because looking at a trial is we might be starting this now, but it might not be ending for another 6 to 10 years, the most critical thing we can do is focus where most of my efforts have been is just to try to coordinate other ideas and to really focus heavily on specimen and data repository, emerging science, and ancillary engagement so that we can make the biggest impact for the most going forward. Because again, it's really critical is that none of these can be validated without the trial. So we have to have an aging outcome trial for validation and qualification as surrogate endpoints. We become this really circular conversation unless we actually just sort of pull together and move this forward. And really important to do that is that we have to have the data, just like those that Morgan has shown and others are really developing, is that we have to support the analytic performance of these biomarkers, showing that they're reliable, showing that they're standardized, showing that they actually mean what they say they mean. And it's going to take a really critical and coordinated effort across investigators to make that happen. In addition to that, we have to have an outcome that we accept as aging. And so I think that that is sort of inherent in what we've been trying to do on the TAME trial and also are trying to approach on some of the harmonized efforts on the smaller proof of concept trials. And really critically, we have to have an effective intervention in order to do all of that together. So it's a tall ask and there's a lot to do. I can post these later for Allison or others, but these are just food for thought. We've been trying to develop, if you did have a perfect biomarker, what would its criteria be? This is not me telling you, this is meant to be sort of a conversation, is that I would love for those that are working on biomarkers that are thinking about trials, is that this is a specific case for a biomarker for a trial of things that we want to see and what else should we be considering so that as we move forward for validation, that we can know if we have the perfect biomarker in front of us, what does it need to do? Okay. And that's all I've got. We've learned a lot. there's been outcomes, there's been pain, there's been tears, there's been 6 years worth of grant writing. There has been a lot of work. And I think one of the biggest points is that, aging reflects a collection of disease conditions, functional measures, biomarkers. We're really trying to capture this larger, broader accumulated deficit, looking at improvements. And this is going to, of course, mean composite measures and aggregate scoring. And I have an army to thank, only some of those who are represented here.